Well, good afternoon. I'm André Drent. I'm the professor in plant pathology at the Center for Horticultural Science and team leader for crop protection. Can you please move to the next slide?
Is that not going?
Now, before we start the seminar, I'd like to acknowledge the traditional owners and their custodians of the lands we meet today. And on behalf of the traditional owners, I pay respect to their ancestors and their descendants who continue cultural and spiritual connections to country. We also recognize their valuable contribution to Australian and global society. They have, after all, been here about 260 times longer than Western society has been here. A few housekeeping rules. The seminar will go from 12 until 1 p.m. sharp, and that includes questions. Now, if you have questions, you can type them in the Q&A box. Please do not put them in the chat box. I will read out the questions at the end from the Q&A box. Now, today we are lucky because we actually have 3 speakers today doing a tech team exercise. So the first one is Associate Professor Elizabeth Dann. The second one is Dr. Akila Prabhakaran, and the third one is Montana Hickey. And they're going to talk about investigating the threats to our green gold, the avocados. Now, the avocado industry has grown a lot over the last decade, and to make this happen, a lot of work is happening behind the scenes. Now, today you'll get a little bit of a glimpse how that is working, who is doing what, because Elizabeth Dann has been part of that journey to enable the avocado industry to thrive over the last decade. So, Elizabeth, the floor is yours. Please start.
Yeah, thanks very much, Andre, and thank you everybody for attending the seminar today. So you might be wondering about our green gold. Where did that come from? So I hope you recognise that the new marketing and branding campaign for avocados is Our Green Gold. It's got nothing to do with the Mexican drug cartels. I know that that's also called green gold, and they're fighting and killing themselves over avocados in Mexico. Not here. And what perfect timing for this marketing campaign to come out. We've all been enjoying the Olympics over the last few weeks. And look, our Olympians did a fantastic job, and they were aptly rewarded with their avocado medals. So it's fantastic to see the avocados really prominent on those podiums. Some of those avocados are not quite the best quality, but that's unfortunately sometimes the experience. And Jess Fox there, she's not biting into her gold medal. She's actually biting into some avocado and cheese melted on Turkish bread. So that's our green gold and where it came from. I hope you're enjoying this season's avocados. There's plenty of them and they're a good price, so please get into them. So panicle and shoot blight and dieback of avocados. Because we're a tag team, I thought I'd just give a little bit of a rundown of who'll be presenting what. I'm going to give a very brief background to the problem, and then Akila's going to take over and she's going to be talking about some work that she started off doing in 2019, looking at the symptoms, looking at some isolations that she did from symptomatic material, and then some fungicide screening work. Montana's going to take over then, and she'll be discussing her honours project from 2020. She also did some isolations from material that we got from samples that year, and she's going to discuss her pathogenicity tests. Then I'll come back at the end and talk about some field trials and some non-pathogenic complexing factors that we think are involved. So very briefly then, around spring in 2019, I got some phone calls and also some emails from growers and agronomists, particularly in the southeast Queensland district. So this is Bundaberg Childers area. And they rang up and they emailed with photos and saying, oh, you know, we're seeing this big problem in our avocados this year. Can you come and help, and can we send some samples down for you to investigate? So in a lot of these blocks, it was a significant problem, and some of the yield reductions were almost 100%, less in other blocks. People sort of observed anecdotally that it had been seen in previous years, but in this particular year, 2019, it was very severe. The Hass and Maluma seemed to be the varieties that were most affected, and the problem was less severe in the Shepherd, the green skin variety. It was more common in determinate flower panicles. So the determinate panicles are these ones here, or functionally determinate. They don't have a vegetative flush coming from the ends of those flower panicles like the lower slide. So that seemed to be a fairly common thing. And at that stage, no one was 100% sure of the cause and thought it might have been related to tree stress. So I'm going to hand over now to Akila. She's going to go through her work. Thanks, Akila.
Thank you, Liz, and good afternoon, everyone. So, as Liz suggested, said in her introduction, the panicle blight or dieback, although it has been observed in the previous years, only in 2019, many samples with, especially the symptomatic panicle samples, were sent to our lab for diagnostic. You can see these pictures of some of the samples we received, taken from some of the samples we received. The first two pictures you can see, this is the typical symptom of panicle blight. You can see in this picture, the symptom initially appeared on the flower. Then the flower along with the pedicel turned black. Then the flower then dropped from the peduncle. Then the whole inflorescence along with the peduncle, rachis, everything turned necrotic, like this one. Especially when panicle blight occurred during the early stage of flowering, the elongation of even this panicle is prevented and forming like witch's broom-like symptom on the panicle. So these two pictures shows the symptom of panicle dieback, which occurred during the later stage of flowering. You can see in the third picture, the panicle is wilted and dieback clearly extended downwards in the panicle. And most of the cases, the dieback arrested at the nodes. But occasionally, like this one, the dieback extended further down to the rest of the branches as well. So these are some of the pictures we received from the growers from different regions at the end of the flowering season in 2019, which shows the impact of panicle dieback in avocado orchards. You can see in the second picture, the impacted panicle doesn't produce any fruit, just leaving the skeleton of the inflorescence panicle. It's also a good source of fungal inoculum, which can be spread to the fruits or other healthy tissues of the tree when during the favorable condition comes. And the third picture, it shows the panicle dieback in the young tree. Apart from the older trees, the young trees also frequently had panicle dieback symptoms, especially when they are trying to attempt the first year of economical yield. So around the same time, in the same year, we also received some samples with leaf blight and also shoot blight. Although the leaf blight is not that economically important like a panicle blight, but again, it can be a good source of inoculum. You can see it can be spread to especially the fruits. The shoot dieback, it's another dieback issue we have in Australia, especially in the new plantings when the trees are less than 12 months old. The shoot of vegetative and the floral shoot have a dieback symptom, and eventually which leads to the death of the tree. Mostly it occurs when the young tree, which are less than 12 months old, when they allow the young tree to the flowering, it couldn't cope up the requirement of the nutrient and water which need for the young tree. So without the proper root development, when the trees are only 12 months old, they couldn't cope up with the requirement of nutrient that put extra stress on the tree. That also could play a role in the dieback, shoot dieback. So similar problems have been observed in overseas, especially in California, South Africa, and Israel, and also in New Zealand. I didn't put the pictures here, but in 2019, when Liz went to New Zealand, she collected many pictures with the panicle blight symptoms. So they also have the same problem. It was suggested that the fungi, Jothriella, belongs to the family Botryaceae, play a major role in causing the problems in avocado in those countries. Apart from the avocado, the similar symptoms also observed in other fruit crops such as mango and lychee. In lychee, the flower panicle and the fruit blight is mostly caused by Alternaria alcinata. And in nut crops such as macadamia and pistachios also had the same flower problem. In macadamia, the dry flower symptom is caused by the fungi Pestilotiopsis. And in pistachios, they have shoot and panicle blight issue, which is mostly caused by the fungi Neophysicoccum. Again, it also belongs to the family Botryaceae. So, to our knowledge, there are no published report or any literature available of panicle blight or dieback in avocado. And at this point, we are not sure what are the pathological and non-pathological factors associated with this problem. So in order to investigate the pathological issues related with the panicle blight, we collected a sample from 14 orchards around Australia. So most of the samples were actually sent to our lab for diagnostic. Among those, more than 75% of the samples were from Childers and Bundaberg region of Queensland. Rest is from Alton Swale from northern New South Wales, and also some samples from Manjimuk, Western Australia. So from the collected sample, isolations were performed with wide range of tissues, as mentioned here. And among the 522 tissue samples we isolated, more than 93% were from symptomatic tissues. So this picture, it's actually botanically the determinate inflorescence of avocado is called as panicle. You can see this depiction was created by our student Montana, and which shows the different botanical terms of the different parts of panicle. So alongside, you can also see the real avocado panicle with severe blight symptom, and also the leaf, old leaf with blight symptom. So these are the different parts which I isolated from. So coming to this slide, this graph shows the different main fungal pathogen group we isolated from symptomatic and asymptomatic tissues. Out of the isolation, total isolation, nearly 40% Alternaria was isolated from more than 40% of the tissue sample, and followed by Coletotrichum species from nearly 20% of the tissue. And Fusarium and Botryaceae were isolated from less than 10% of the tissue. And there were some others and unknowns, which was nearly 15%. The others were like Pestilotiopsis and Cladosporium, and to some extent Epicoccum. Some bipolaris were there. There was some no growth were observed on some 6% of the symptomatic and asymptomatic tissues. So this graph shows the different fungal pathogens isolated from different tissue types. Again, you can see the green bar, which represents the Alternaria species, was isolated very frequently from all the different type of tissues we isolated, and followed by Coletotrichum species were isolated from leaves and also some extent from peduncle and stem. In contrast to the anecdotal reports and the other published reports on other crops, we have isolated very less frequency of Gaucher's Periaceae. So among the three major isolated fungi, Alternaria and Gaucher's Periaceae fungi are not a new one to avocado. They are well-known pathogens in avocado, especially the Coletotrichum species, which is well-known to cause anthracnose on the fruit surface, and also the Gaucher's Periaceae fungi, Neophysicoccum and Laceo. They are well-known to cause stem end rot, and also the dieback in avocado as well as other tree crops. However, we don't know much about the pathogenicity of Alternaria in avocado. In general, Alternaria can be here as a saprophytic fungi or even primary pathogen. We don't know. And however, we know that the Alternaria can be a primary pathogen in many crops. It's reported to be causing leaf spot and rot as well as blight in more than 300 host species, especially the Alternaria solani, which is well-known pathogen in potato and tomato. So, so with this one, we don't know whether the pathogenicity of Alternaria in avocado at this point, and also we didn't have flowers at the time to immediately test the pathogenicity of these isolated fungi in avocado panicle. So from here, instead of waiting to test the pathogenicity of these isolates, we decided to do the in vitro fungicide screening in order to find the suitable candidates to include our field trial. So we did this in vitro screening by using the Petri plate assay. You can see on the right corner, she is Yuping. She's a visiting scientist from Taiwan. She was here with our avocado plant pathology team for 6 months and very actively involved with in vitro screening along with me. So we both made nearly more than 2,000 plates to complete this in vitro screening project. And so Liz had a very brief discussion with many major fungicide company to choose the important good fungicide candidates to include in the in vitro screening. The good ones, like the ones potentially in the advanced stage of development, or the ones which are already registered in the other crops against the similar pathogens. So after thorough discussion, we chose 10 chemical and 5 biological fungicide to do the in vitro screening. So the fungicide were tested at 5 different concentrations, starting from 10 milligram of active ingredient per ml up to 0.001 milligram of active ingredient. And the EC50 value was calculated for each fungicide product. However, because of the method we adopted to do this assay for the biological fungicide, we couldn't calculate the EC50 value. But you can see in the pictures here, some of the new chemistry showed significantly higher inhibition to the frequently isolated pathogen Coletotrichum and Alternaria, even at a very low concentration, 0.1 milligram per ml. So for these fungicide, we did repeat the test with further lower concentration, up to 0.001 milligram per ml. So this table basically shows the sensitivity of fungicide to different variation in sensitivity to different fungicides. And in the test in vitro screening, this is the result only from the chemical fungicide. As I said earlier, we didn't include the result of the biological one. We included the copper fungicide and also some of the old, that is the fungicide, which has been used in the avocado orchards for many years, and some of the new fungicides such as Lunar Sensation. And these are all new fungicides, some of them, as I mentioned, which are not registered in avocado or in the final stage of its development. So based on the EC50 value, that EC50 is nothing but the effective concentration in which the 50% of the, which required to control at least the 50% of the fungal growth. So based on the lower EC value, EC50 value, we selected these 4 fungicide candidates, 3, 4, 5, and 6, to include in our field trial. So the field trial results will be presented by Liz in the later part of this talk. Thank you for listening. If you have any question, I'm happy to answer at the end of the talk. Now I'll give the talk to Montana to talk about her pathogenicity trial results. Thank you.
So thanks, Akila. So I then followed on from this project that has been done so far. And in 2020, I started my honours to investigate the pathogenicity of these fungi associated with panicle blight in avocado. So these symptoms did come out again in the 2020 flowering year. So and this was some of the symptoms that we saw. And for my honours project, I created 2 research questions. And these were, are fungi associated with panicle blight in avocados? And if so, what are they? And the second question that I created was, do fungi associated with panicle blight cause disease in asymptomatic avocado flowers? So these were then formulated into the hypotheses. If one or more fungi are associated with panicle blight in avocados, the causal agent or causal agents will be able to be isolated on selected media from symptomatic samples. And to follow the Koch's postulates, the second hypothesis was that if a fungus associated with panicle blight is pathogenic, then it will cause disease symptoms after inoculation, which can then be re-isolated. So to actually do this, I then first studied the terminology of avocado panicles. And as Akila said earlier, the main 4 parts that I was focusing on were from the stem, the rachis, the peduncle, the pedicel, and then the flowers. And what I did first was some preliminary experiments in both glasshouse and lab experiments using cultivars Haas, Maluma, and Shepherd. And the main point of these was to develop a method for the later pathogenicity experiments and also to create a scale for scoring the process. So from there, I also did a sample collection in Queensland orchards, and I took small pieces of tissue, plated that on nutrient agar, and incubated them for 7 to 10 days or until sporulation, so that I could identify those fungi morphologically, and any of those cultures of interest I then stored. And later on, we chose 6 of those cultures as fungal candidates for the pathogenicity experiments. And so there were 8 treatments all up, a water control, these 6 candidates, and a sterility test, which was a second water control at the end, which was used to ensure that my sterilization techniques were effective. And so I did 2 of these trials, each lasting 7 days on detached panicles. And these were with Duke 7 cultivars, and each of these panicles had at least 6 flowers per panicle, and there were 9 panicles per treatment. So all up 72 panicles for each of those 2 experiments. So from there, I then re-isolated fungi to check the Koch's postulates, and I took 6 flower pieces, 6 pedicel pieces, and a rachis piece per panicle, and then inoculated, put those into nutrient agar and incubated them to identify those fungi morphologically based on the representative isolates that I stored and inoculated the asymptomatic panicles with. So from that, I then did molecular identification of those candidates using ITS 4 and 5 and TEF 1 and 2 when appropriate to identify those two genus so far. So for my sample collection in 2020, most of the sites were in Southeast Queensland and then another from North Queensland. And there were 15 samples that we received. And from there, I did 551 isolations from symptomatic tissue. There were another approximately 300 isolations from asymptomatic tissue, but I haven't included that for this presentation, as they mainly just either had no growth or fungi that we didn't expect to be pathogenic. So from those 551 isolations, 128 had no growth at all, so 23%. And the remaining 423, we did find fungi that were able to be identified, and that will be shown on the next slide. And out of all of these, Alternaria, again, was the most frequently isolated at 22%. So this was really interesting, as Alternaria has been found in areas where avocados grow in previous years and decades, but it has never been found to actually cause disease on avocado. So it's interesting that we were isolating so much Alternaria. And I'll show you on the next slide, and you can point these ones out, but the main candidates that were identified from the sample collection were Alternaria colotochicum species, Pestalotiopsis, which later was identified as Neopestalotiopsis, and then also Nigrospora. So you'll see that on this slide here, where the green bars indicate the symptomatic tissue pieces, and the yellow bars represent the symptomatic panicle tissue specifically. So that was those 4 pieces that I said earlier with the rachis, peduncle, pedicel, and the flowers. So on that left side there, the Alternaria is the highest there with the 23%. And then on the far other side, on the right side, there's the other column, which is also quite high. But that consists of all the other species that either were not able to be identified as they were smaller, more insignificant ones, or other fungi that were able to be identified but were in such small quantities that we didn't expect them to be a candidate for this. So the main other ones that are of interest here are the colotochicum, which was the second most frequently isolated at 16%. And as Akila said earlier, this does cause stem end rot and anthracnose in avocados currently. And then from there, the next highest was that Nigrospora on the right side, and Pestalotiopsis were some key ones there. I did later select to use a species in the Botrytis family, even though you can see third from the left there, it is the lowest frequently isolated group of fungi, but we did receive a sample from North Queensland, from Kathy Grass, who sent us a sample of Lasiodiplodia theobrome from panicle blight symptoms. And because of its current effect as a serious pathogen in avocado, we then decided to include that as well in our experiments. So I mentioned this scale earlier that I developed, and this was a scale of necrosis that I used to score the symptoms. So this ranges from the 0 to 6, 0 being 0% necrosis, to 5 with 100% necrosis, and 6, 100% necrosis, with those symptoms then spreading to the peduncle or the rachis, whichever was appropriate. So these numbers here were then converted to a Likert scale and analysed with the Crystal-Wallace and Dann tests for significance. You can see that here, these are the results from the 2 trials that I conducted, and the main 5 species that were significantly different to the control are indicated with the red line above the graphs. So this was the colotochicum, Alternaria species 1, Alternaria species 2. So I used 2 of those as there were 2 distinct species that we found, the Lasiodiplodia theobrome and Neopestalotiopsis. So Nigrospora was the only candidate that was not significantly different to the control and the sterility tests. So from our reisolation then, we had some interesting results here with the I used the mean reisolation percentage here, and this was analysed with the beta regression. And Colotoxicum, Alternaria species 1, Lasiodiplodia, and Neopastoloideopsis were all significantly different to the control and sterility tests. And something that's interesting in this graph here is that Alternaria species 2 had quite a low reisolation percentage in comparison. And this we thought perhaps could be because Alternaria species are known to produce mycotoxins, which may not require the fungi to actually infect the plant at such a high percentage, but mycotoxins can cause necrosis in plant material. So perhaps this is how that Alternaria species 2 had such a high necrosis score, but a lower reisolation result there. And so overall, I answered my first question of whether fungi are associated with yes in the species that I've been speaking about. And to answer whether they can cause necrotic symptoms, the greatest necrosis was caused by Lasiodiplodia theobrome, Colotoxicum, Alternaria species both 1 and 2, and Neopastoloideopsis. And the most frequently reisolated ones reflected that necrosis score, except for the Alternaria species 2. So the main future work for this would be to conduct implanted pathogenicity experiments, as these were with detached panicles. And the other way is going to be looking at future management strategies, which Liz is about to speak to you about now.
Great, thanks, Montana. Okay, so going on to what we've actually done field-wise to look at this problem. So without really knowing whether there was a fungal cause or not, we did set up 2 fungicide spray trials at Bundaberg Childers area last year. And we selected those 4 candidates that Akila mentioned from the in vitro testing and compared them with nil control and also the industry standard control fungicide program and a couple of other treatments that I won't be going into today. So there were 2 fungicide sprays during flowering. Pretty much all avocado growers won't spray any fungicides at flowering, but we were able to show that there were no phytotoxic effects of those 2 fungicide sprays on the flowers. So that was one of the key concerns with the fungicide, with the avocado growers, is the potential risk of phytotoxic effects. Kayleen Bransgrove is also in my team, and she came up with me a couple of times, and we collected some pre-spray panicles and also post-spray panicles with small, tiny match head sized fruitlets, and she's done some isolations from those, and that's a story for another time. There were no effects of the fungicide treatments on panicle dieback when I assessed those trials in October. So it didn't seem to be that the fungicides were controlling or managing panicle dieback. We also tagged hundreds of flowering panicles with flexible tags and counted them 3 times from September through to December. So you can see the picture on the right shows one of those panicles which has small fruitlets develop. Avocado flowers profusely, but only a very small percentage of those flowers actually set fruit and hold to final yield. So there were indications of effects of these fungicide sprays on fruit retention during October to the December period where we were counting these panicles, and also on yield when yield was assessed in June 2021, so June this year. So the lower left graph, you can see that one of those fungicide treatments, the red one, actually had higher numbers of fruitlets per panicle pretty much right through until December period. And that also translated to a high yield in June. So the other fungicide treatments or nil control had very, very low numbers of fruitlets per panicle right through that flowering and fruit fruit incision period. That was also reflected in very low yields from those treatments. So this whole block where I tagged those panicles from 2 rows of trees and approximately 120 odd trees, we only got a few bins of fruit from. So the problem is still having a significant impact on yield. We've also collected fruit from the yields in June, and that was assessed, ripened and assessed for post-harvest disease, and we're still going through the data for those post-harvest assessments. So then, visiting the field so often, I wondered, well, you know, is it something that we can prune out? So in November, I took a red spray can and actually spray painted some trees. The grower was kind enough to let me have 12 trees outside of my trial rows, and I spray painted where I wanted the farm manager to go back and cut those trees, which he did, and painted them white to protect those trees from sunburn. So you can see that in January, there's not much growth yet. But in June 2021, there's already quite a lot of regrowth from those pruned trees. So it'll be great to go back in a few weeks' time, hopefully, and just touch base with those pruned trees to see if they're flowering. And again, to track those 12 trees through flowering to see if they develop panicle dieback and to see if they've got a useful yield. So also talking to various growers and farm managers about a potential plant nutrition effect. So here really looking at what can be some of the non-pathogenic factors that might be involved with the complex. I selected about 40 leaves from several different trees. Some of those trees in highlighted green were what I considered healthy trees with very minimal panicle dieback. Other trees had significant panicle dieback, and they're in the orange highlighted boxes. So overall, the calcium percentage and the magnesium percentage were about 20% lower for the panicle blight trees, and this is in leaf analyses in February. The potassium was nearly 30% increased in those sick panicle dieback trees. So then that sort of makes us question, well, is plant nutrition a cause or a consequence? And I tend to think it's probably more a consequence of panicle dieback, and suspect that it's more likely related to a weaker root function, so less root volume or limited function of those roots, so that those trees are not taking up moisture and nutrition. So again, a potential non-pathogenic factor, and it's something that growers mentioned. So Bundaberg Childers, in particularly in 2019, had a very, very dry, extended drought period. So even though these trees are irrigated, they are potentially stressed. So if you can follow the dark purple bars in 2019, looking at the average monthly rainfall compared with the mean in the green bars. So right through in 2019, June, July, August, September, October, the rainfall for those months is significantly lower than the mean. In 2020, the rainfall was more likely or more average for those key months, except in October, where the October rainfall was about half of that of the average. And we're just tracking that through now for 2021, and already it's either around average or July much, much higher. So we'll wait and see what happens as we go through October, September, August, September, October. So some work that my friend and colleague Tony Wiley did several years ago, he's an avocado physiologist. He showed that in Fuerte cultivar, the surface area of flowers is almost the same that of the total canopy, so that the leaf area during peak flowering. So this means that flowers are a significant source of water loss through the canopy. It's also likely that in Bundaberg Childers area, we know that water is quite saline as the dam levels drop. that water becomes even more salty. And it's not uncommon to see leaves with this salt burn on them. So just wrapping up for the whole presentation then, I think there's two phenomena here, possibly they're interrelated. The panicle blight, as far as the yield loss is concerned, I think is less of a problem than panicle dieback. However, we are receiving a sample that's probably going to come in today from North Queensland, and I think the photos that that agronomist has sent me show definite panicle blight, a fungal cause for flower blight and necrosis. So it's ongoing. Panicle dieback is widespread, and I think it's a complex interaction between environmental, physiological factors. Not sure whether there's a fungal cause to it or not. Alternaria and Cortotricum are consistently isolated at very high frequencies. As Montana mentioned though, and Akila also, Alternaria is very common saprophyte, and you can isolate Alternaria from just about anything. Whether or not it's really a problem in this case, it's yet to be determined. Montana's work show that fungi can cause flower necrosis, and her work showed that Lasiodiplodia, which is one of the Botrytis is highly pathogenic. So further work there is necessary with intact attached flowers. Hers were detached flowers, and potentially even expanding the range of fungi that we test in that pathogenicity, further pathogenicity work. Fungi vary in their sensitivity to fungicides, and that's not unusual. Some of the newer generation fungicides seem to really effectively inhibit most of these key fungal genera that we see in avocado. We're seeing some positive effects of some of these new fungicide candidates on yield and in fruit quality. That's some of our work that we've just been processing that data from. And we're actually going to establish a second field trial in the Bundaberg or Childers area, and that's got to commence fairly soon because already some of those trees are starting to go into some early flowering. The dieback skeletons, as we call them, are indeed a significant source of inoculum for further fruit infections. So the skeleton, or the dried, desiccated, blackened panicle that you can see on the top right, is covered with fuzzy, furry, grey spores and mycelia. And this is a sort of inoculum if it rains and then washes down onto the fruit surface and cause what we don't like to see, and that's anthracnose on avocado fruit. So this really has implications for canopy management and general orchard hygiene to get those sources of inoculum well away from any developing fruit. So I think we've made good progress on this issue in the last couple of years, and we've got a little way to go, but I think we're honing in on some really interesting and potentially some answers for this issue. So thanks everybody. I'd like to move forward. Finish by acknowledging our trial collaborators, Auschili and Simpson Farms. We're very grateful to have them let us do our trials on their orchards. Also, their farm staff have been fantastic and very accommodating. Syngenta, Bayer, New Farm and Tanuki all provided product for testing and have great interactions with those companies. Mitch Faint we employed as a contractor to do the spraying so that we weren't running up and down the Bruce Highway to do spraying. And very big thanks to my whole pathology team. And finally, the project is funded through Hort Innovation and uses the Avocado Grower Levy Fund. So thanks very much for that funding source. And now I would like to open it up for some questions. Thanks, Andre.
Okay, well, thanks very much, Liz, Akila in Montana, for your presentation. Excellent job. Yeah, there are already several questions, and it's quite interesting because I can see where they come from. and who has them. And there's quite a few questions from people from other states, so that is a good sign. Let me start with a few simple questions first to get you sort of warmed up to the thing. Are there any issues to do with resistance in varieties? Is there a difference between these diseases between different varieties?
Yeah, we don't know. So the problem seems to be worse in Hass and Maluma, less severe in Shepherd. Whether or not it has escaped because it's an earlier flowering variety, we don't know. Maluma is an early variety also, and it does seem to be affected. I'm not aware of any reports of there being a true resistance amongst cultivars to this or any other flower or panicle problems.
Now, is there also a relationship between the age of an orchard in, let's say, especially Alternaria, because that tends to build up in older plantations, you know, you get more inoculum, so it starts very small. And then did you check the age of the orchards where you get most of the problems?
We did, and at first it seemed to be predominantly in young orchards, which was a real surprise to me. And I know Akila showed one photo that we took in northern New South Wales, and as she mentioned, it was their first commercial yield really, and I've seen the same thing in southwest WA. So it's one of these things, the more you're familiar with it, the more you actually see it in the orchard. Yeah. We're seeing it in young and older orchards.
Now, is there, Tim O'Hare had a question, whether or not determinate panicles are more susceptible than indeterminate ones?
I think the determinate panicle thing comes down to more the tree stress. So determinate panicles probably are more common in stressed trees, and potentially that relates to why we're seeing it more associated with those determinate panicles. So already the tree is stressed, it's already not producing so many indeterminate where the flush comes out. Okay. So, yeah.
There's a few people, they have asked some questions about, you know, which pathogen. So I'll just try to get a few. One of them is David Guest. So he has a question whether or not you consider the possibility that a pathogen is an obligate biotroph. So did you actually also isolate from dead tissue in advance of the lesion to see if actually some biotrophs there, and whether or not you actually did DNA diagnosis well in advance of the necrotic lesion?
I'm going to hand that question over to you.
That's a good idea.
Yeah, thanks. Can you please repeat the question, Andre?
Yeah, did you consider the possibility that the pathogen is an obligate biotroph? So basically, something you cannot culture on a plate. So did you then, to check that, did you look in advance of the lesion by DNA test if there is a pathogen there?
No, we haven't done that test, but we just did only some molecular characterization of what we isolated, what are the fungal we isolated. We haven't done any molecular characterization of those things. So,
and did you sample also then from non-symptomatic as well as from symptomatic tissue?
Yeah, that's the problem with the first year sample in 2019. Most of the sample we received were symptomatic. So we didn't go on, we collected only few samples. Those samples, when we went and collected, we collected along the asymptomatic sample as well. Whereas the sample we received from the growers, most of them were symptomatic. So we didn't have the good number of samples to compare versus asymptomatic and non-asymptomatic tissues.
Yeah. And there's another question related to the pathogens, and that is whether or not bacteria could be the cause, because something like pistachio dieback is caused by a Santamona species. So did you actually look and try to isolate bacteria?
We didn't do separate bacterial isolation. most of the isolation we did in the PDA plates. But when we have bacterial looking pathogen, we just checked with the other people, but we didn't have much bacterial isolation from the tissues we isolated. Maybe I can hand over to Montana to just let her explain whether did she get anything in 2020.
Hey, so my project did focus on the fungal aspect, so we mainly looked at that. And I didn't actually get any bacteria out. There was only one time when we got it out, and it was one thing, but we thought that was an issue more with the plates rather than the actual sample. And we mainly focused on looking at the actual symptoms on the panicles and actually looking at it. It gave quite a number of symptoms that related to fungi. And we wanted to have a look first at that to make sure that we could target that if that was the cause. There definitely could be future research into looking at whether there is bacterial or different organisms that we can't culture or anything else that could be in the environment. Same as why we're looking at the different environmental stresses, because there could be other reasons as well. So this is kind of the first step, and we're mainly just looking at the main clues that we've been given to direct us in where to look first.
There is another question which follows up from that. That is whether or not that are two pathogens working synergistically. They don't have to be fungi causing this problem.
Yeah, absolutely could be a potential cause. There could be definitely more than one, and I guess this first stage of determining what is there is what we've been focusing on, and then a number of the pathogens that or number of fungi that I was looking at, some of which are pathogenic to avocado already, some which aren't. They're definitely around in the environment. There are so many endophytic fungi and organisms, so there definitely could be a mixed interaction, could be something to do with timing or stresses. So I guess that's very well potentially a reason, but yeah, this is kind of where we're at this stage. We're giving you as much as we know at the moment.
Yeah, okay. So no seminar would be complete with a few questions about the control of the disease. So, is there, if new fungicides are introduced, do they have an effect, or are they safe for pollinators? Have you looked at that?
That's the other major concern of avocado growers is, yes, fine that they're not phytotoxic on the flowers, but do they affect the bees? The chemical companies are the ones that have that data, and they would not let me or anybody put on their fungicides if they weren't happy that they were safe for the pollinators, which are really, really important. So I've not gathered that information about whether there was an effect on the pollinator visits or effective pollination, but one of the labels, and this is one of the copper compounds that's already registered for use, the label actually says apply at flowering and then through the season. So you can apply them at flowering, but that work specifically has not been done by my group. I hope it's been done by the chemical companies.
Yeah, but there is sort of another question related to that is, people say fungicides work or they don't work, but the timing of application for these sorts of diseases is critical, because you don't actually know exactly when infection takes place or where the inoculum is building up. So what is have you looked at that? And what is your sort of view on that? Where do we go from here with this stuff?
Yeah, I think, I'm in two minds about whether to start a fungicide program in the next trial. So to use some curative type fungicides which have some not protective, but they actually get in and knock out infections that have already happened. So come in early with one of those before flowering or at the very early stages of flowering, and then again potentially at either later flowering or early fruit set. The main problem, as I've mentioned, didn't seem to be with blight at flowering. It was well after that. So hard to know which way to take it. But I think There are some benefits in using a fungicide at flowering.
Yeah, okay. We are moving up to 1 o'clock. So can you please move to the next slide?
Yep.
So if you enjoyed this seminar, next week we have another seminar, and that is Jack Christopher. And he's going to talk about increasing wheat yield resilience to late season drought by targeting stay green traits in root architecture. I just will translate that title for you. How can a wheat grower squeeze a little bit more yield out of the tank? I think that's sort of what it's going to be about. It's going to be an interesting seminar, how you can get there. Now, I'd like to make a few closing remarks. Thank you very much for attending the seminar today. If you enjoyed the seminar, you can actually do a little poll at the end of the seminar. And I'd like to thank Liz, Akila, and Montana for an excellent seminar. Thank you very much. Bye.
Thanks, everyone. Thanks for attending.
Thank you.